Hi, my name is Kumar Alagramam. I'm an associate professor and director of research at the University Hospital Ear, Nose, and Throat Institute, University Hospital, Cleveland Medical Center, Case Western Reserve, University School of Medicine, Cleveland, Ohio. I'm going to talk about small molecule and gene therapy approaches to mitigate hearing loss in Usher syndrome type 3. Usher type 3 is characterized by postlingual progressive hearing loss, a variable presence of balance disorder, and variable onset and severity of retinitis pigmentosa, which leads to loss of vision. Mutation in the Clan1 gene is linked to Usher type 3. Clan1 gene codes for Clan1 protein, which is a membrane protein as depicted in the lower right corner of the slide. Please note the yellow molecule attached to the extracellular loop of Clan1. This sugar molecule is attached to amino acid asparagine, single letter code N. I will talk a bit more about it in the next slide. The two common mutations in Clan1 are Clan1 Y176X, also referred to as a fin major mutation. The other common mutation is N48K. The amino acid asparagine, single letter code N at position 48, is replaced by lysine, single letter code K, therefore N48K. This mutation is common among Usher patients of Ashkenazi Jewish descent. To have an idea what these two mutations means, I depicted them in form of the key shown in the bottom of the slide. In one case, the N48K, the protein is made, but it's unstable. In the other case, the Y176X mutation introduces a premature stop, resulting in premature translational stop or a truncated protein. This is similar to a broken key or a null effect. Some symbols that you might encounter in the slides are CLRN1 representing Clan1 protein, HL hearing loss, delta symbol for mutation or change, and squiggle to represent similar to. Why hearing loss happens with Clan1 mutation? So I'll talk about that using the mouse models. I'll then move on to the small molecule we developed to mitigate hearing loss associated with the N48K mutation. The second part of my talk will focus on gene therapy to preserve hearing in the Clan1 mutant background. When I refer to mouse models, I mean mice carrying mutation in Clan1 and showing hearing loss. These mice or these mouse models do not develop eye disorder. We do not know why. The focus of my talk will be about hearing loss and mitigation of hearing loss associated with Clan1 mutation. More than 10 years of research compressed in few slides that you're going to see, therefore the details are kept to a minimum. If you have any questions, please write to me at kna3@case.edu. This slide shows human anatomy compared to the mice. Particularly, the focus is on the human organochordae, which is in the cochlea. The main cells are the sensory cells in the organochordae, the rows of inner hair cells and outer hair cells. This structure is identical to the structure in the mouse. Therefore, the mouse provides a good model to study hearing and deafness associated with hair cell defects. Why do we call them hair cells? The sensory cells in the ear have hair-like projections on the apical surface, therefore the nickname hair cells. The hair cells convert sound vibration or vibration from the head movement into electrical signals, which are then conveyed to the brain via the auditory nerve or the vestibular nerve. Any defect to the hair cell, genetic or environmental, will result in hearing loss. So why hearing loss with Clan1 mutation? What do the mouse models tell us? To the right of the slide, the top panel shows the scanning electron micrograph of hair cells from a normal mouse. You see that the hair bundles are organized as an inverted V shape, and they are arranged in a neat fashion. And the bundles are organized. The hair bundles are organized. When Clan1 is mutated, the hair bundle organization is disrupted, as seen in the bottom right panel. The hair bundle is the mechanosensory structure of the hair cell. Therefore, mutation in the Clan1 results in hearing loss, as shown in the panel to the left. The graph in the panel to the left shows hearing of the normal mice at the bottom. See the green line? These mice can respond at about 35 decibel sound intensity. What is 35 decibel equal to? Approximately sound intensity of a whisper. In contrast, the null mice, Clan1 null mice shown in the top and red line, does not respond even to 100 decibel sound intensity. 100 decibel sound intensity would be the sound you hear at wood shop. The N48K mice do show some degree of hearing at earlier stages. This is indicated in the blue line in the middle of the plot. However, they quickly lose all of the hearing. The bottom line of this slide is loss of Clan1 or mutation of Clan1 affects hair cells, which in turn results in loss of hearing. In this slide, I'm showing in vitro studies to understand why Clan1 N48K is dysfunctional. The top panel shows cells that express normal Clan1 protein fused to a reporter, a GFP protein. The proteins do not have any fluorescence of their own, so fusing it with the reporter is a standard laboratory technique. The top panel shows the normal Clan1 protein locates or goes to the cell membrane. The bottom panel shows a similar experimental condition where the Clan1 N48K protein fused to GFP is expressed and observed. First, Clan1 N48K protein can be seen in the cell only if we add chemical agents to block protein degradation. Second, we notice that the Clan1 N48K protein is stuck inside the cell. That is, it does not reach the membrane. So these are two critical pieces of information. Combined with the in vivo data that you saw in the previous slide in the mice, we hypothesized that increasing the stability of Clan1 N48K would help the mutant protein reach the proper site in the cell and rescue Clan1 mediated function in the affected cells. Now, proteasome inhibitors can increase Clan1 N48K stability in cell culture. But we cannot use these as drugs for disorders like Usher3 because it is toxic. Therefore, we need to find small molecules or some other drug that can specifically protect N48K version of Clan1 and assist the protein to reach the membrane. In this slide, I show the front page, the first page of our recently published paper in Nature Chemical Biology. The title of the paper is "A Small Molecule Mitigates Hearing Loss in a Mouse Model of Usher Syndrome Type 3." This was published April 25, 2016. I have boxed all of the authors, I have put a box around all of the authors' name to emphasize the point that this is a team effort. If you need more details about the small molecule therapy for Usher Type 3, please look up this paper. This slide shows essentially a schematic of our effort to screen a small molecule library consisting of about 50,000 small molecules. Here, cells expressing the Clan1 N48K protein fused to a reporter gene is screened against each of those 50,000 small molecules. After a couple of years of hard work, we arrived at one compound which is labeled O03. This compound stabilized Clan1 N48K, and it was found not to be a proteasome inhibitor. So it was stabilizing Clan1 N48K, and a portion of the protein was seen at the membrane in the presence of this small molecule. So this molecule, O03, is a lead compound. The structure of this is shown in this slide to the left, and at a concentration of 2 micromolar, the EC50 was 2 micromolar for this lead compound. However, we would like to bring the dose of this lower. That is, we would like to lower the EC50 while we keep the potency the same. Medicinal chemists were involved for more than 2 years working with this compound to come up with the most ideal or more optimal small molecule labeled and shown here to the right, which is BF844. So this compound stabilized the N48K and assisted some of the protein to reach the surface of the cell. This is in cell culture, and it could do this at a much lower concentration than the lead compound, as indicated in the EC50 below. So we have now, thus far, at this point in our work, we have obtained a small molecule that is able to stabilize the N48K mutant protein. The next big question is whether this small molecule can preserve hearing in the mouse model with the N48K mutation. So this slide shows simply the schematic for an auditory brainstem response, one of the tests used to check the hearing sensitivity. The top right start with a sound provided to the ear. The ear then provides a signal to the auditory nerve, which is then passed on to the brain, and from the brainstem you have an electrical response which is recorded by subdermal probes that are placed in the subject's head, and the hardware connected to this recorder converts that information to a waveform as shown in the bottom left panel under normal mouse. So this is the waveform you get when you have a normal hearing subject. The numbers shown here, 46, 51, 56, shows that increasing intensity of sound. As the sound intensity increases, you see the amplitude of the waveform also change. So in contrast to the normal mouse, if you look at the lower middle panel, the Usher3 mouse at about 100 days essentially shows no response whatsoever, even at the highest intensity. Basically flat line. That means the animal is profound and displays deafness. The right lower panel indicates what we expect would happen if a particular treatment is successful. We will essentially restore those waveforms. This slide shows the actual test of the small molecule in the Clan1 N48K mice. The orange diamond, the bottom, represents response of a normal mice in ABR tests. So these mice can respond at about 35 decibel sound pressure level. What does that mean? That sound intensity is equal to a whisper. In contrast, the blue square shown at the very top represents the results from the N48K mice untreated. Even at 100 decibel sound, a sound that's similar if you're standing in a wood shop, that's how much sound you would get. Even at that sound, these mice do not respond. When you treat the N48K mice with the small molecule, the results are dramatic. This purple triangle shown in the middle, 3 purple triangles in the middle, indicate that the hearing in the N48K mice treated with a small molecule has significantly improved. How significantly? Our calculations show that it is 10,000-fold improvement. To summarize the first part, we have developed the first targeted therapy for an Usher syndrome, particularly for the hearing loss. BF844 preserves hearing in Clan1 N48K mouse. Since the Clan1 N48K causes both hearing and vision loss, BF844 administered systemically could in principle prevent both sensory deficits, that is the vision loss and hearing loss, in patients with Usher syndrome type 3 associated with the N48K mutation. This slide summarizes all of our effort in the small molecule project. At the 12 o'clock position, we start with the N48K subject and going down to the 2 o'clock, 3 o'clock, 5 o'clock, 7 and 9 o'clock position, where we end up showing efficacy of the small molecules, like using the mouse model in vivo. So that's how far we have, and at this time the Usher3 initiative has taken the small molecule BF844 and they are proceeding with additional preclinical studies that are necessary for regulatory approval. And hopefully in the future we will be able to take this to clinical trials. I'm going to shift gears and go to a different, my second part of the talk. This is more exciting development, more recent. It's gene therapy tested in Usher3 mouse model. This paper was published 18th October 2017 in the journal called Scientific Reports from the publishers of the journal Nature. The title of the paper is Modeling and Preventing Progressive Hearing Loss in Usher syndrome type 3. Again, all of the authors are, and I put a box around to indicate contributions are many, and this is a team effort. So, what is gene therapy? The normal copy of a gene is replaced, transplanted, if you will, into the mutant cells of the target organ to correct the genetic disorder. Why gene therapy for Usher type 3? Especially after I talked about the small molecule. Now, small molecule BF844 in this case won't work for all mutations in Ushers. For example, the Y176X, essentially no protein is made. 844 is only useful if there is a mutant protein present and with a particular type of mutation. Whereas gene therapy will enable the synthesis of Clan1 protein irrespective of the nature of the mutation. Then you can ask the other question, why did we bother developing BF844 for Clan1 and 48K if gene therapy looks like it might work? In case of the Clan1 and 48K, a functional protein is made, but it needs help to keep up, to deliver Clan1 mediated function. BF844 assists the mutant protein to do so, but if you don't have any protein, BF844 won't help. The second thing is, in case of the Clan1 and 48K, BF844 can reach both the eyes and both the ears and treat all sensory organs at the same time, whereas in gene therapy you have to treat one eye and one ear at a time. So the advantages of the small molecules, it depends on the nature of the mutation. So in this slide I'm showing the mouse model that we developed in the knockout background of Clan1 with a progressive hearing loss. The bottom line, the black line, represents the response from the normal mouse, so these can respond at about 25-30 dB, whereas The Clan1 knockout, shown in the red at the very top, fails to respond at the highest intensity of sound presented. The blue line represents the new progressive hearing loss model in the Clan1 knockout background. So earlier on, at 22-30 days, it shows relatively normal hearing, more to the left of this graph, marked by P22. As with more time, the hearing gets worse, as indicated in the blue line going at nearly 45-degree angle. So the new model shows delayed onset progressive hearing loss in the Clan1 null background, whereas the knockout mouse alone, the older model, showed early onset profound hearing loss. So we used the new model to test our gene therapy because this progressive hearing loss is similar to what is seen with the Usher3 patients. This slide shows the Clan1 gene therapy construct that is used with the AAV virus viral gene therapy vector. So the cDNA, Clan1 cDNA, is in the middle of the construct, and this viral gene therapy vector was injected into the ear of mice that are 2 to 3 days old. And after that injection, we wait for about a month and look at the hearing over a period of time. So this is similar to what I showed before in terms of the hearing test that was done. One point I want to emphasize is that this is very similar test done in humans as well. So we are using comparable tests to determine the efficacy of either the small molecule or the gene therapy. So this shows the actual results of the gene therapy that we did. The black line in this graph represents the hearing of normal mice, which is not treated with anything. And these mice can hear at about 35 dB. And the hearing is sustained. The x-axis shows the days from day 27 to day 150 when the study was stopped. The blue line in this graph represents the progressive hearing loss model with Usher type 3. Earlier on, the hearing is around 40 dB, and slowly hearing sensitivity decreases, and you need to provide more and more sound, and by about 90 days, even at the highest intensity of sound provided in this test, these Usher3 untreated mice fail to hear. So these are the ones with the knockout background. The red line is what you need to focus on. This is the progressive hearing loss model. This is the Usher3 in the knockout background. This is the new mouse model. And this, when the AAV virus is injected in the ear of these mice, they seem to sustain their hearing over a period of time. The AAV viral gene therapy has prevented the progression of hearing loss compared to the untreated mice, and the hearing sensitivity is stable over a long period of time, as shown here, until 150 days when we stop the study. So what this data shows is that gene therapy with Clan1 gene is very effective, and when the viral vectors are delivered before the onset of hearing loss, you can see hearing preservation is robust. Conclusion of part 2, we have developed a progressive hearing loss mouse model for Usher3 in the knockout background. We have developed a gene therapy approach that is very effective in curtailing progressive hearing loss in the Usher mutant mouse, and the effect remains stable. Gene therapy vectors were introduced very early, i.e. before the onset of hearing loss in this model, and that seemed to be very effective. The next step in gene therapy for Usher3: will gene therapy work in the new Usher3 mouse model if the viral vector was introduced in adult mice? If so, how well? And we also would like to test gene therapy in Usher3 mouse model using new generation viral vectors such as ANC80. And hopefully, if everything goes well, apply for regulatory approval for a trial. So the papers that I described, these two yellow, links, hyperlinks, can take you to the two papers. They are free, and if you're interested in more information, you can go there, and also you can send me an email if you have questions. I would like to thank Usher3 Initiative for funding this project, and some of the support also came indirectly from NIDCD. And also I would like to thank University Hospital, Cleveland Medical Center for financial support as well. And with that, I'd like to thank everybody. Thank you very much.